Thanks to the fortuitously freeze-dried skin, scientists have successfully accomplished the groundbreaking task of reconstructing the chromosomes of a woolly mammoth that lived 52,000 years ago, marking the first ever instance of such a feat. Greetings, everyone! Today's video focuses on an incredible achievement by a team of international researchers. They have managed to reconstruct the three-dimensional structure of chromosomes from woolly mammoths that lived 52,000 years ago. This remarkable study has unveiled fascinating information about the genome architecture of these ancient creatures, revealing that they had 28 chromosomes similar to modern elephants. Through the analysis of gene activation patterns, the researchers have also identified genes associated with woolly traits and cold tolerance. This groundbreaking approach has the potential to revolutionize the field of ancient DNA studies and may even contribute to the de-extinction efforts of woolly mammoths. Without any further delay, let us dive into our discussion. According to Erez Lieberman-Aden, the director of the Center for Genome Architecture at Baylor College of Medicine and the corresponding author, this fossil represents a novel category and far surpasses the size of individual fragments of ancient DNA by a staggering million-fold increase in sequence. Moreover, this marks a groundbreaking achievement, as it is the inaugural instance of establishing a karyotype for an ancient specimen. The wealth of information obtained from understanding the three-dimensional structure of a genome extends far beyond its linear sequence. However, the challenge arises when dealing with ancient DNA samples, which typically consist of fragmented and disordered genetic material. Inspired by previous research on mapping the 3D architecture of the human genome, Aden hypothesized that locating a suitable ancient DNA sample, one that retains the intact 3D organization of its fragments, could potentially enable the assembly of ancient genomes using similar methodologies. After conducting extensive testing on numerous samples for a span of 5 years, the researchers finally stumbled upon an exceptionally well-preserved woolly mammoth that had been unearthed in northeastern Siberia in 2018. According to Olga Dudchenko, the corresponding author from the Center for Genome Architecture at Baylor College of Medicine, they believe that the mammoth underwent a spontaneous freeze-drying process shortly after its demise. Dudchenko further explains that the nuclear structure within a dehydrated sample has the remarkable ability to endure for an astonishingly extended duration. In order to recreate the genomic structure of the mammoth, the scientists obtained DNA from a sample of skin located behind the mammoth's ear. Employing a technique known as Hi-C, they were able to identify the regions of DNA that are likely to be physically close and interact with each other within the nucleus, reflecting their natural state. According to Marc A. Marty-Renom, a corresponding author and structural genomicist at the Centre Nacional d'Anàlisi Genòmica, CNAG, and the Center for Genomic Regulation, CRG, in Barcelona, envision a scenario where you are faced with a puzzle consisting of 3 billion pieces, but you lack the image of the final puzzle to guide you. However, with the utilization of Hi-C, you can obtain an approximate representation of that image prior to embarking on the process of assembling the puzzle pieces. Using the genomes of contemporary elephants as a framework, researchers merged the findings of the Hi-C analysis with DNA sequencing to construct a systematic blueprint of the mammoth's genome. Through this process, they were able to identify the specific sections of DNA that interacted with each other. Surprisingly, the analysis uncovered that woolly mammoths possessed 28 chromosomes, mirroring the chromosome count of present-day Asian and African elephants. Even more astonishing, the fossilized mammoth chromosomes retained an impressive level of physical integrity and intricate detail, including the intricate nanoscale loops responsible for facilitating communication between transcription factors and the genes they regulate. Through an analysis of gene organization within the nucleus, the scientists successfully distinguished between active and inactive genes in the skin cells of the mammoth, serving as a representation of epigenetics or transcriptomics. Notably, the mammoth skin cells exhibited unique patterns of gene activation in comparison to its closest kin, the Asian elephant. These distinctive patterns potentially correspond to genes associated with the mammoth's ability to withstand cold temperatures and its characteristic woolly coat. According to Marty Renom, we now possess woolly mammoth tissue that provides insight into the specific genes that were activated and deactivated. This groundbreaking discovery offers a completely novel form of data, as it represents the initial instance in which cell-specific gene activity has been measured in ancient DNA samples. Despite relying on exceptionally well-preserved fossils, the researchers remain hopeful that this method has the potential to analyze various ancient DNA samples, ranging from mammoths to Egyptian mummies, as well as more recently conserved specimens found in museums. To further advance the study of mammoths, the next course of action would involve analyzing the epigenetic patterns of various other tissues. According to M. Thomas Gilbert, a paleogenomicist at the University of Copenhagen and the Norwegian University of Science and Technology, these findings bear significant implications for ongoing endeavors focused on the de-extinction of woolly mammoths. To support our channel's growth and ensure broader awareness, kindly hit the like, share and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.